All right, hi everyone. Today I'm going to talk about center surround suppression and how it's been studied in a couple of mental illnesses. So mostly depression, and mostly I'll talk about depression, and I'll also talk a little bit about schizophrenia. So remember, we talked about center surround suppression earlier in the course when we talked about retinal ganglion cells, right? So those are the cells where you've got a circle, right? And if anything comes within the circle, the neuron is going to fire. But it's a little more complicated than that, because really only in the center of the circle do things increase the firing rate of the neuron. In this wider area around, called the surround, if something shows up there, it actually decreases the firing rate of the neuron. That's called center surround suppression. It's how many neurons in the visual system work. And it's cool. And we've learned that from single cell recording studies. Remember, I showed you guys the Hubel and Weasel video where the cat was looking at the screen, and they would have little wires in the back of the cat's head, and they'd record the activity of the neuron that they happened to be next to, because it was connected to an amplifier. They could hear what the neuron was doing live, and they'd show it stuff and get it to figure out how it worked. And one of the things they figured out is that some neurons, particularly in the optic nerve, these neurons that are connected to the retinal ganglion cells, or that they are retinal ganglion cells, the axons of the retinal ganglion cells, if you put a wire next to those, a lot of them have a center surround organization. In other words, wow, if we show the cat this little teeny spot, it fires a lot. If we show the cat a really big spot, it actually fires less. Okay, so that's a review of center surround suppression and what it is. So there's people are interested in how that might be altered in mental illness. And it's tough, because what are you going to do? Crack open someone's head and shove a wire in there and start measuring to see whether receptive fields are, they have the same center surround organization, or maybe they're like, think of all the ways that it could be altered in a mental illness. Like maybe the center is a little extra small, or it's a little more sharply tuned, that it increases its firing rate a lot for just like less of a stimulus or something, or it's slower to increase its firing rate. There's so many ways it could be altered. But unfortunately, in general, we don't study humans with single cell recording. The rare exception is if someone's having brain surgery. Sometimes they'll allow researchers to put a little wire next to their exposed brain, because they're getting brain surgery anyway and removing a part of the brain, and they poke around the brain as a matter of routine when they're doing that, so they don't like remove the wrong part of the brain. They're kind of checking around to see what different parts of the brain do. But anyway, usually we don't do that. We don't record single cells in people's brains. So what we can do is there are certain outcomes from center surround suppression, from that organization of the individual neurons, that play out at a higher level. So if you put a bunch of those neurons together, like 1 million or whatever, that all have a center surround organization, the outcome is basically that the perception of an object that's relying on millions of neurons also sort of has this center surround organization to it, or center surround effects can be observed in people's perception, basically in their performance. If you do a psychophysical task, you can show, oh, look at this result. I can only explain it based on some sort of center surround interaction that's going on, okay, in receptive fields. So this is what I'm going to talk about today. So first we have to understand that result, that center surround result that we can observe not from individual neurons, but from human beings' performance on a task. So I'm going to show you a paper about that. Okay, so here's the paper I'd like to show you that sort of demonstrates how center surround organization plays out in people's perception. How does that affect people's performance on a task? The fact that our neurons are organized that way, that center surround organization of receptive fields. So this is about visual motion processing, which is a good area for you guys, because you already know about what that is. It's this. A lot of the studies in visual motion processing are using these grating stimuli, these squiggly lines that move left or right. And you saw that in the Yu Chen paper, the 1999 Psychophysical Isolation of Velocity Discrimination in Schizophrenia. So we talked about that task a bunch. So this is the same task basically. You look at this grading, these wavy squiggly lines here, and they're going to either drift to the left or they're going to drift to the right, and you're going to say which way they go. So that is the task that people are doing in this study. Now this study is in Nature, which is, top journal in the world probably, of all science, Nature and Science being right at the top there, those two. And to be, just a perception study where you're just doing a task on a computer, and to get into Nature, it has to be a really big deal. So this paper is a really big deal. A lot of people cite this paper now. This came out in 2003, and it's been pretty influential in at least this part of the world of motion processing. So the main thing you need to understand that one of the reasons it got into Nature is they did a lot of cool things in this paper, and they show it all with these different graphs and stuff. You don't have to worry about all this. I'm just going to show you basically two results that'll be good for you to understand. So the first result is this. So both results are shown in figure 1A here. And let's see if I zoom in to that. Yeah. And there. Now you'd be less distracted. Just oh, it doesn't want to. There we go. Figure 1A. So the first result is this. So remember I told you you're going to see which way the lines are drifting. And in psychophysics, we always want to modulate the difficulty of the task somehow so that we either acquire someone's threshold or we get a curve of people's performance across different levels of difficulty. And that's what they're doing here. They're modulating the difficulty of the stimulus to acquire someone's threshold. So remember, the threshold is just like, if I had a light bulb and I wanted to see how good you were at seeing brightness, I would put different voltages through the light bulb, like 1 volt or 2 volts. And maybe I find out that you need 3.5 volts of voltage to be running through that light bulb for you to be able to reliably tell that it's actually on versus off. That would be your light bulb threshold. And what they did in this task for motion processing is, if you just look at it, drifting lines, you can tell which way they're going, left to right. It's not too hard. But to make it difficult, the thing they modulated was how long did they show the lines to you for. So if they only show you those lines drifting to the left or the right for 1 millisecond, I mean, you can't tell. Your brain needs longer than that to be able to pick it up to say which way it was going. So that's what's shown here is the duration threshold. They measured how long do you need. I'm going to give you a bunch of trials, and we'll see how long you need to be able to tell which way the wavy lines are going. And so this is one condition here that they measured someone's threshold. And for right here, this little dot, what this means is that it took someone about 90 milliseconds, see it's right before 100, to be able to tell which way the dot was going, and whatever condition this is. This other condition here, they needed probably about 50 milliseconds to say which way the wavy lines were going. So that's what the y-axis is. Great, we understand that. Now what's the x-axis? Well, the x-axis is how big the stimulus was. So remember, this is the stimulus, and you could make the wavy lines have a different radius or in diameter. They could be really small, or they could take up this whole circle, or they could take up the whole screen if you wanted them to. In this study, they chose 5 different sizes. They did 0.7 degrees of visual angle, all the way up to 5. Now, a degree of visual angle, I may have told you this in class, but a good rule of thumb to remember how big a degree of visual angle is, if you hold your thumb out at arm's length, 1 degree of visual angle is about the width of your thumbnail. So that's about how big the stimulus was, a little smaller than that for their smallest condition. And then their biggest condition, it wasn't absolutely huge. I mean, it was 5 thumbnails. Okay, so that's what we're talking about in terms of the different sizes they used in this study. Is 1 thumbnail all the way up to 5 thumbnails, basically. Now, let's take a look at one of these lines. It's so confusing and overwhelming if you look at this graph. How am I going to understand it? Well, the way is one thing at a time. So we look at just one graph here. Let's look at this. I don't even know which one this is, but it's this black circle graph, okay, line. Let's look at that one. So we already saw that 90 milliseconds is the threshold. That's how long you need to see when it's a very small stimulus, okay? What happens when I make the stimulus bigger is that the threshold goes down and down and down and then stays about the same down. So as you change the size, this becomes easier or harder to see. The threshold is going down, so what does that mean? It means that it's getting easier to see, because if your threshold is lower, it means you need to look at it less long. You can tell quicker which way it's going. So people are better and better at this the bigger the stimulus is, okay? Now that makes sense, right? Because if a stimulus is large, like my grandmother can't see too good anymore, and so she has playing cards with really big letters on them. It's easier to see stuff that's big. It's easier to see an elephant than a gnat, right? So it sort of makes sense that if you have a bigger and bigger stimulus, you do better and better with it. Okay, so that's cool. Well, that's this one here, this one line, okay? Now let's look at another line. Let's look at this line here. Now here you start at 35 millisecond threshold, and as you make it bigger, it goes higher and higher and higher and higher, all the way up to 130 milliseconds you need when it's a really big stimulus. Now what is going on with that? That means that as you make this line, whatever this 92% means, that this line, as you increase the size of the stimulus, it gets harder and harder for people. They need more and more time. Well, that's the exact opposite result we were just talking about with this first line we looked at. So this diamond line and the circle line are the opposite results. Now that's messed up. Must be something, whenever you get results like that are completely the opposite of each other, I hope you have a good explanation for why that is. That would be really exciting. But in this study, they do have a really good explanation for why that is. And that's why the study is in Nature, is because they say, wow, with this line, I make it big and they do better. With this line, I make it big and they do worse. Well, what's the difference between those two lines? 92% refers to the contrast level of the stimulus. So that means, if you look at this thing, it's pretty high contrast. Matter of fact, I'm going to show you another paper that demonstrates this contrast a little bit better. They didn't have space in this paper because it was in Nature. Nature doesn't give you a lot of space. Instead, we'll go to one of my papers, which was not in Nature. Okay. You see? Here, this would be a high contrast stimulus, and this would be a low contrast stimulus. Can you barely even see it, right? And this would be a high contrast small stimulus, and this would be a low contrast small stimulus, which again you can barely see. Let me double the zoom. So you can see that's very faint, and this is very high contrast. Now here, this is high contrast, and this is very, very faint. You can barely even see it. And these guys, more so than the figure you just saw in that other paper, these give you a sense of what it looked like on the screen. So this is actually more or less the whole screen, what a computer monitor size would be, and this is the size of the stimulus, whereas this is a bigger stimulus here. Okay, so going back to this, high contrast, they do worse when you make the stimulus bigger. Low contrast, you do better when the stimulus is bigger. And why is that? Well, center surround suppression is the answer. So remember, when we have a stimulus and we stimulate the surround area, it decreases the firing rate of a neuron. Well, if you do that to a lot of neurons, you decrease the firing rate by stimulating the surround, you're going to mess up the signal that the center is trying to tell you, which is trying to tell you the answer of which way it's going. And so that's why people do worse with a big stimulus, is because you're getting a lot of surround activity suppressing the signal of which way the thing is going. This does not occur at low contrast. So this is something I never taught you before, but with single cell recording, if we record a a cell, but we're showing it really low contrast stuff, like stuff the cell can barely even detect, center surround suppression does not occur. Instead, the bigger you make the stimulus, the more the cell will fire. Center surround suppression only occurs for high contrast stimuli. Okay, so that's why people said this result has to be from center surround suppression. I mean, look at it. High contrast stimuli are worse when it gets bigger. Low contrast stimuli are better when it gets bigger. It's got to be from center surround suppression. These threshold differences have to be a result of that. There's no other way to explain it. Usually, a big stimulus is easier to see. So the fact that a big stimulus is harder to see at high contrast has to be from center surround suppression. Okay, so that is the interesting Taddin. This is a paper by Doohy Taddin. He's a guy who went to Vanderbilt. He studied under Randy Blake. Randy Blake is the guy who wrote the textbook that you guys read, your vision chapters earlier in the year, Blake and Sekula. So this is a very good research group here. So since this paper came out in 2003, a bunch of people have tried to study how does center surround suppression, as we measure it in visual motion processing on a task, how is that altered in mental illness? And a bunch of people have looked at this. I'll just tell you about mine. If you're interested, there's others. There's actually someone named Julie Galome who studied this in depression like I did, and she found the opposite result as me, more or less. And there's people who studied it in schizophrenia. But for now, I think I'm noticing my video is getting kind of long, so I'm just going to talk to you about my study. So, what we did is we studied, it's called dysregulation of visual motion inhibition in major depression. And what that basically means is I did the same thing that Taddin Nature paper did, and I studied it in depression, and I found that it was, somehow altered. And one of the arguments I made in the paper in the intro is about this GABA issue. So, how does center surround suppression work? Do you guys remember I was telling you guys about the motion detectors and how do they work? And well, maybe it's because 3 retinal ganglion cells are hooked up to this motion detecting cell, and they have different amounts of myelination, and so the signal reaches the cell at the same time, even though it started at different times when an object was passing through a certain area. I drew a little fly on the board passing through 3 receptive fields, and the 3rd receptive field has the fastest myelination, and the 1st receptive field has the slowest, but he starts going, and so they reach the motion detector at the same time. And remember I said I was kind of talking out of my butt there. I didn't really know if that's how motion detectors work, but it's probably something like that. Well, okay, so here's another example of, well, how does center surround suppression work? Why is it that when you stimulate the surround area, it decreases the firing rate of the neuron? Well, my sense is that no one really knows for sure how that works, or at least not a lot of detail on how that works. And one thing that might be involved is GABA neurotransmission. So what all that means is some sort of action potential where a neuron is firing and sending neurotransmitters to another neuron using the GABA neurotransmitter. And the reason for that is GABA is the primary neurotransmitter that's used in your brain when your brain is trying to suppress something. Anytime one part of the brain is suppressing another, it's using GABA to do the neurotransmitting. It's not using dopamine, it's not using serotonin, it's using GABA. So the interesting thing about depression is that when people are depressed, they have less GABA in their visual cortex. This is just a study that came out a while ago. The only reason they measured the visual cortex, no one cares about vision except us in this class, right? And a few other people. The only reason they measured the visual cortex is because it happens to be in the back of your head. And for technical reasons, when you're in a brain scanner, you can measure the back of the head pretty well to see how much GABA is in there using something called magnetic resonance spectroscopy. It's like using an MRI machine to measure the concentration of a chemical in the living brain. It's pretty cool technology. So people found that people who are depressed have less GABA in the back of their head, in the visual cortex. And so, okay, for us, we say, wow, center surround suppression probably relies on GABA neurotransmission. There's less GABA in the head when you're depressed. Shouldn't depressed people have less center surround suppression? Shouldn't they have less visual motion inhibition, is how I phrased it in this paper, but I really mean, shouldn't they have less center surround suppression? If that doesn't make sense, rewind 10 seconds and watch that again. That logic should make sense to you based on what you know from the class so far. So that's what we were interested in. This is me explaining center surround suppression to the reader, which you guys already sort of know, but this was our task. We basically had a small or a big stimulus, and we did a little different than Tadden, that other Nature paper. We didn't measure the threshold. What we did is we just measured the accuracy at different presentation times. So something like this. So here you see for a big stimulus, well actually let's look at this one. This is the best one to start with. Oops. So for a small stimulus, high contrast small stimulus, this is how long we showed you the stimulus for. And this is your accuracy level. So obviously, for a short presentation time, you're not good at it, and you're not good at it again, but then you start to get better when it's shown for longer. And once you show it for over 100 milliseconds, people can do it quite well. So that makes sense. And what we found, not to our surprise, as expected, was that depressed people could do this just fine. Nobody thought there was anything wrong with visual motion processing in depression. So we were sort of happy to see that, wow, people with depression can do this just fine. They can tell which way this little stimulus is going. But remember, we're interested in center surround suppression. We think that might be messed up because they don't have enough GABA in the back of their head, people who are depressed. So we looked at the big stimulus where center surround suppression is presumably going to affect your performance. And what did we find? Well, I drew the people with depression have the blues, so they're blue. And healthy controls are gray. And so you see that healthy controls are going up with longer presentation times, and so are people with depression, but they are actually kind of skewing down here. And let's think about it this way. If there were, if you look at this graph, do people with depression have more or less center surround suppression than healthy controls? Well, if they had less center surround suppression, they would actually do better on the task in the large condition, because you can't really tell from this graph, but look at like this here, 66 milliseconds, that's right around between the second and the third little dash for accuracy, okay? Now if you look here, the third, 66 milliseconds, it's not even to the second dash. So even for controls, they are lower accuracy for the big stimulus here. So if you have less center surround suppression, you should be better in this condition. And that's not the case. What we found is that people with depression really stunk at the big condition. The controls stink at it too. Or they don't stink, but they're worse at the big condition than they are at the small condition. People with depression are even worse at the big condition than the small condition. They're not impaired with the small, but with the big, they're super duper impaired. And that was the opposite of what we thought we would find. Because remember, we thought we'd see less center surround suppression, which would actually make you do better on the big condition. So this is really interesting and confusing. This was a group of people who were depressed who did our study. Someone else had done a study before on this, and she did it the way Tadden did. She measured threshold. She didn't measure the accuracy. So there's a little bit of a methods difference that might explain part of the results, but she also studied people who had recovered from depression. She didn't study people who are currently depressed. She did study people who were depressed in the past. And she found that they did have reduced center surround suppression, as everyone would have thought from the GABA literature. I studied people who are currently depressed, and I found they had too much center surround suppression. So that is an interesting difference. It sort of helps, I think, if you, in my paper here, if you just subtract the accuracy in the big condition from the accuracy in the small condition, then you can see the difference. And that difference is sort of the center surround suppression level. And you can see for controls, it peaks around 66 milliseconds, how much center surround suppression there was. And for patients, it peaks around then too, but it's more. They have more of a difference here. Now I'll tell you one more thing because this is sort of interesting, which is that medications didn't really affect the results. So this is all depressed people, and you can see people on meds did have a little bit more surround suppression than people not on meds, but it was kind of the same pattern and not too much of a difference. I wouldn't make too much of this. There's some other stuff here with low contrast stimuli that I won't have time to talk about, but if you're interested, shoot me an email. We can talk about that. So yeah, that's sort of the state. Is this with this depression stuff? Is it's this situation where there's something going on. The people with depression are performing differently with regard to center surround suppression, but there's conflicting results in the literature, and I got a different result than the other person who studied it in people who weren't currently depressed but had recovered from depression. So this is an area where there's some more work to be done to kind of figure out what's going on. So good area to think about.